Chemistry of carbohydrates. Uh, Under this part, we shall define carbohydrates, classify them, give their properties, and so on. So carbohydrates are organic substances made up of polyhydric alcohols and an active aldehyde or ketone group. They supply the largest part of the energy needed by the living cell. From this definition, it is obvious that two kinds of carbohydrates are possible: those containing an aldehyde group and those containing a ketone group. Examples of both kinds can be seen in the formula of glyceric aldehyde and dihydroxyacetone. The simplest carbohydrates are called sugars. It can be seen now that two kinds of sugars exist: the aldoses, those which contain the aldehyde group, and the ketoses, those which contain a ketone group. Our introduction to cell biology will end by briefly looking at a few points on gene expression, but it will not be discussed in its entirety as that is part of genetics. So the genetic code is the basis of DNA information. DNA information is organized in information units called genes. A gene can be defined in many ways. It is usually interpreted as a functional unit of heredity, consisting of a segment of DNA located in a specific site of the genome. Each organism has a characteristic number and complexity of chromosomes. Chromosomes are thread-like collections of genes and other DNA in the nucleus of a cell. The term chromatin is used to describe chromosomes that are being used to run differentiated cells. Genetic engineers are skilled at alternating gene information coded in the DNA. In addition, certain biotechnology applications involve making artificial genes by synthesizing strands of DNA that are then placed into cells. There are three major types of code in the DNA programming. Amongst these are the regulatory DNA, the structural DNA, and the junk DNA. So all these have functions. The function of regulatory DNA: first, it is composed of chromosome segments and whole genes that function to regulate the expression of other genes. Then structural genes carry the code for structural polypeptides and enzymes that build other structural components of a cell. And then junk DNA is a common type of genetic information that either has no definitive role or helps in reducing the effects of environmental factors that damage DNA. Certain types of junk DNA provide flexibility in the genetic code. This type of DNA is regularly exploited in biotechnology investigations involving metabolic engineering. Gene expression is composed of two stages. The first stage, which takes place in the nucleus, is called transcription. This stage copies particular sequence of DNA to fulfill a cell's needs. Every three sequential nucleotide bases in the DNA molecule form a code to match a specific amino acid, and thus each trio or triplet of bases is known as a codon. For example, the DNA code ACC programs for the UGG rather codon. This codon is the information from amino acid tryptophan. The order of codons is a section of DNA determines the amino acid sequence in a protein. The copied segment of DNA derived through transcription is a nucleic acid known as messenger RNA or mRNA. The next stage of gene expression is called translation. Translation takes place on ribosomes located either in the cytoplasm or the ER. The process of translation uses mRNA to direct the synthesis of proteins from amino acids.